Hello everyone, welcome back to Psychopharmacology. This week we're doing Chapter 3, Chemical Signaling by Neurotransmitters and Neurohormones. First off, we're going to talk about chemical signaling between neurons. Okay, so let's start off by talking about some of the basics of communication between neurons. Synapse means gap. It's the point of communication between neurons. So as the presynaptic neuron makes a connection with the postsynaptic neuron, they don't physically touch. There's a small gap on the order of 20 nanometers that we call the synaptic cleft. That is the area where neurotransmitters are exchanged from presynaptic to postsynaptic neuron. So we'll use the terms presynaptic and postsynaptic a lot when we're talking about this kind of thing. It's a relative term, right? Relative to the synapse. So the neuron that is releasing neurotransmitters across the synapse is the presynaptic neuron, and the neuron that's receiving on the other side of the synapse is the postsynaptic neuron. These aren't absolute terms, right? Like most neurons are both presynaptic to the neurons that they're talking to, and they're postsynaptic to the neurons that are talking to them. So when we say pre and postsynaptic, it's in reference to a specific synapse. The postsynaptic density is the area of the dendritic membrane that's facing the cleft that contains many neurotransmitter receptors. So that's basically the hot zone where all of those neurotransmitters are going to come in and be able to find receptors to bind to. Postsynaptic density. So there's a few different types of synapses that we can talk about. The first is an axodendritic synapse, which is a synapse between an axon terminal and a dendrite. This synapse often happens at spines, dendritic spines being those sort of swollen portions of the dendrites, which are specialized to receive communication. We have pictured here from an electron microscope such a connection, an axodendritic synapse. We can see here dendritic spine, so the portion of the dendrite which is receiving the communication. Here we have our presynaptic terminal. You can see contained within the terminal are synaptic vesicles, so there's little sacs that contain neurotransmitters. You can see here in this dark portion the synaptic cleft, the site of communication between neurons. So in addition to the more common axodendritic synapse, we also have axosomatic, which is a connection between the presynaptic axon terminal and the postsynaptic cell body. So instead of making contact with a dendrite, it makes contact with a cell body. There's also axoaxonic, which is what it sounds like. Presynaptic axon terminal making contact with a postsynaptic axon terminal. So it's terminal-on-terminal connection. What's usually happening here is the presynaptic neuron is altering neurotransmitter release from the postsynaptic cell directly at the terminal. So terminal A is synapsing onto terminal B. It can affect the release of neurotransmitters from terminal B. This can either reduce through a process we would call presynaptic inhibition or enhance through a process we would call presynaptic facilitation, the neurotransmitter release from that terminal. In addition to neurons, there are other non-neuron targets. For example, neuromuscular junctions that allow us to contract our muscles, or contacts with cells that can release hormones as part of our endocrine system, also targets for neural communication. So here we have pictured the three types of synaptic connections that can occur between neurons. We have our axodendritic, where the terminal of the presynaptic neuron is synapsing on the dendrite of the postsynaptic neuron. For axosomatic, we have the terminal of the presynaptic neuron synapsing on to the soma of the postsynaptic neuron. Then axoaxonic, you can see here, we have the presynaptic neuron synapsing onto the terminal button of the postsynaptic neuron, allowing presynaptic facilitation or presynaptic inhibition, altering the amount of neurotransmitters released by this neuron here. Here's a figure from your textbook that summarizes the major categories of neurotransmitters. We'll talk about each of these categories and in fact many of the examples at much greater detail as we move through the course. So we have the classical neurotransmitters, the amino acids such as glutamate, small neurotransmitters here, monoamines, including dopamine, norepinephrine, serotonin. We have acetylcholine, which is kind of its own thing. We have purines. We'll talk a bit about some of those later on. Then we have the non-classical neurotransmitters, like the neuropeptides, which are large molecule neurotransmitters, which means they get created and processed a little bit differently. We have lipids and gases, which are super weird in terms of how they are synthesized and released. But we'll talk about all that in just a moment. So as we mentioned, neurotransmitters, the chemical messengers of our nervous system, are typically one of the following. They're classified as one of the following: amino acids, monoamines, neuropeptides, lipids, or gaseous. And we'll talk about what each of those means in more detail as we move on to talk about those specific types later on in the course. It's worth noting that neurons can often co-release multiple neurotransmitters. So just because a neuron produces dopamine, let's say, it doesn't mean that's the only neurotransmitter that it produces or releases. So we can have a co-release of multiple neurotransmitter types from a single terminal. You can see here we have small vesicles containing classical neurotransmitters, which are smaller, and large vesicles containing these larger neuropeptide type neurotransmitters. Okay, so most neurotransmitters are made in the terminals. They can be synthesized locally. Enzymes that do that work are created and trafficked from the cell body, so they're made up here and then trafficked down using your neuron's transport system. But that's not true for every type of neurotransmitter. So while our smaller neurotransmitters can be made near the synapse and packaged into vesicles there, that's not the case for larger things like neuropeptides. So neuropeptides are made from protein precursors, which are synthesized here in the cell body. So precursors are made in the cell body, and then they must be trafficked down to the terminal. So they're assembled up here, packaged, and then transported down the axon to the terminal. Then we can have breakdown of these larger protein precursors into neuropeptides. Then the neuropeptides can be fused and released as part of neurotransmission. The action of those neuropeptides is then halted by enzymes that can degenerate those peptides into smaller constituent pieces that can then be recycled. As a result of that, replenishment is slower than for small molecules, right? Because a large precursor protein has to be synthesized in the cell body, then trafficked and degraded, and then sent out. It takes longer to replenish it than smaller, more classical neurotransmitters, which can be synthesized right here in the terminal. In addition to that, we have some other classes of neurotransmitters that don't use vesicle systems at all. They're not packaged into a little sac that has to fuse with the membrane to be released. Some gaseous and lipid-soluble neurotransmitters are just made on demand, so they're made in response to cellular events. And because of their solubility, gases and lipid-soluble neurotransmitters can't remain bound to the membrane, so they diffuse away immediately and do their signaling. So they're made on demand in response to cellular events and are not packaged and stored ahead of time. So there also exist neuromodulators. Neuromodulators are kind of defined by their function that they may enhance, reduce, or prolong the action of a neurotransmitter. So they modulate the effect of a neurotransmitter. These also have the property that they may diffuse away from the site of release, so they can diffuse greater distances and influence other cells. We call this volume transmission. So that sort of diffuse signaling where it can drift away from the initial site of action and have effects elsewhere is called volume transmission, whereas typical synaptic transmission that's confined more or less to a synapse is called wiring transmission. This distinction can be somewhat unclear. So neurotransmitter and neuromodulator can be used interchangeably in some instances. For example, serotonin, depending on where it is and what it's doing, could be considered a neuromodulator. So we're not going to really make a point of making a distinction in this class. If something is serving to modulate activity, I'll talk about it functionally, but I'm not going to ask you to define what is a neurotransmitter versus what is a neuromodulator, because it's kind of nebulous. But we'll talk more about neuromodulation in terms of what is actually happening with a specific system. Okay, that's it for our first mini lecture in this unit. We'll see you next time.